Hello, good morning everyone. My name is Mafalda and I am the team leader of the scientific department of the 8th edition of the Bioengineering Week. Here today I present you the lecture Upstream and Downstream Processing of Antibodies from the cell line to its application, moderated by Professor Raquel Aires-Bach. Thank you all for being here today. You can scan the QR code through your camera, not through the SBE app, so you can earn biocoins and win or great prizes at the end of the week. If you cannot do it right now, I can show it again at the end of the lecture. Don't forget to submit all your questions through the SBE app. So at the end of the session, they will be answered at the Q&A session. Without further ado, I hope you enjoy the lecture. You find it as interesting as we've hoped, and I hand the floor to Professor Raquel Aires-Bach.
Thank you.
Thank you.
So, thank you for the invitation. It's a pleasure to be we are the three guests here that I know for a long time. So my name is Raquel Aires-Bach. So I'm a professor in the Department of Bioengineering and a researcher in the Institute of Bioengineering Biosciences, IBB. And I work for a long time with antibodies and downstream processing and antibodies. So it's a pleasure to be here. I think I have to speak in English, but I propose, is there here somebody that doesn't understand Portuguese? No. So I propose that we do the presentation in English, but then the discussion in Portuguese, I think. Then it's better. So I think the first speaker will be Isabel Campos Pinto. Isabel is a research scientist at the Biosatellite Laboratory at IBET, and she works in life development of next generation sequencing technologies for antibody discovery. Isabel was a former master in biotechnology here in Técnico, and also she worked with antibodies, and she'll present our research in this field. Then we have António Grilo. Is currently working at the Foundation Medicine's Patent Pool, an NGO that jointly with the World Health Organization co-leads the mRNA Technology Transfer Program. So he is working for also some time with antibodies, manufacturing process of antibodies at Lonza. He has a PhD in chemical engineering at Imperial College, and he also has done his master thesis in bioprocess engineering at Técnico. So all of them have some connection with Técnico and with me. And then Cecilia Roque is associate professor in the Department of Chemistry at Nova University, and also researcher at UCBio. She has a degree in chemical engineering and a major in biotechnology from Técnico also. And she has also been working with antibodies and biomimetic ligands, and so is also antibody people here. So now I think all of you are going to do a presentation of 15 minutes, and then after that we'll open the discussion. So the first speaker here will be Isabel, the youngest one. I think it was the order. Then I don't know.
Vocês querem que eu apresente aqui, certo?
Sim, sim, sim.
Isso está para mim. Agora vais para o pé.
Pensava que não sabia se sentada.
Ah,
não, mas isso pode ser
uma cadeira.
Não sei, acho que não vale. Pode ser em pé. Vocês têm algum daqueles tipo ponteiros? Ou
Não.
não só registro? Não, a professora não tem algum daqueles ponteiros.
Não tenho.
Não faz mal. Okay. Okay. Hello, everyone. First of all, thank you for inviting me, and it's always a pleasure to be here at Técnico with Antonio and Professor Raquel and Professor Anne. Raquel. Yeah, and so today my name is Isabel, and I'm currently a scientist at Bayer Satellite Lab in IBET here in well, not in Lisbon, but in Oeiras. And today, with my presentation, I want to give you more of an academic perspective of how it is to work in antibody discovery. I've always worked with antibodies. I consider myself, I would say, like a wet lab researcher and a scientist. I've always worked at the bench, and I've only worked with antibodies. Since I worked here at Técnico, I've only worked with projects that were somehow related with antibodies. So today my idea is to, well, I have an academic perspective. I've worked both in companies and in universities, but I would say I've always worked mostly in like research and academic institutes. So my perspective will be very academic. I know that Antonio will have a different view on antibody discovery field. So I will show you what I've done in the past almost 10 years in this field, and I will show you what probably you will encounter if you decided to work in this area. So for you to have an idea of what I've done in the past years, so I studied biochemistry at Faculdade de Ciências here in Lisbon. Then I moved here to Técnico, where I did my master in biotechnology with Professor Anna and Professor Raquel on the downstream processing of antibodies. Then I stayed here for 1 to 2 years more with a scholarship, and I continued my master thesis work, which was mainly on purifications and antibody purification, so totally focused on downstream processing of antibodies. Then I moved to England, where I did a PhD. It was an industrial PhD, so it was with Aston University, but I was based at a company called Mologic, and Mologic was a company totally focused on the development of diagnostic tests. And I was there for 3 years doing my PhD, and then I stayed for another 2 years as a research scientist, doing as well antibody discovery work for many different projects, and then I will explain you a bit later. And then after England, I went to Denmark, to the Danish Technology University, and I did a short postdoc at a tropical pharmacology lab, which was also in antibody discovery, but for snake venom therapy. So I was working in the discovery of antibodies for improving snake venoming and venoming therapies. I was there for 6 months, and then I got the opportunity to come back to Portugal, and now I'm working at the Bayer Satellite Lab at IBET, which is also a group totally focused in antibody discovery and assay development. And I will talk to you about what I've done. I will talk mainly about my PhD, because I feel it was like the most complete project that I've worked on, from the upstream to the downstream, from generation of antibodies to its application. And then I will also talk about what I'm doing right now at Bayer. So I made this workflow, which I think it's not fully complete, but what I want you to show you is that antibody discovery takes, I don't know, decades, 20 years, 10 years, 20 years. It takes really long time to get an antibody in a pharmacy as a drug therapy. So what I want to show you is that I divided this workflow into phases. So there is a discovery phase, there is a preclinical development phase, and then you still have the clinical trials in human beings. So I personally have only worked in the discovery phase, where I decide what is the target that I want to use. I do the discovery itself, which involves phage display and all the discovery of candidates. And then I also worked a little bit in lead optimization, which involves antibody engineering. I've never done in vitro or in vivo testing with animals. I've never really worked in big pharma companies where they do liters and liters of large-scale expressions, and I haven't even worked with clinical trials. So what I can talk is about the discovery phase. So if you want to find an antibody, you need to start by knowing against what target you want to raise an antibody. Is it a protein? Is it a gene? Is it a peptide? Is it involved in a disease? You need to know what kind of disease you want. In the end, you want to cure a disease, or you want to affect a pathway that is related to a disease. So you need to know what proteins are involved, select your target, and then test if it's feasible to generate an antibody. So that's the first step. Then once you know what are your targets, you start the discovery phase. So you decide if you want to do immunization to get immune response from an animal and get polyclonal antibodies, or if you are going to do everything more in a recombinant way, if you are going to do phage display, you'll do rounds of selections, enrich your libraries until you get a few candidates. It may be 10 candidates, 100 candidates, 1,000 candidates. And then once you have a selection of good candidates, then you start working on them, and then you may do, for example, antibody engineering, trying to make them more efficient with higher affinity, with less immunogenicity. And those were the things that I've worked on in the past years. So I have this slide. I don't know if it's just the basics of antibodies. And I'm sure you probably have learned this in classes. There's polyclonal antibodies, monoclonal antibodies. I'm just going to explain more or less this because this was the basis of all my works. So polyclonal antibodies, they come from different populations of B cells, so they bind to different epitopes on the target. Monoclonals, they come from a single population, and then they bind to a specific epitope. And then there's all, of course, other differences. If you want to just generate polyclonal antibodies, it's just easier and faster in a way. So you just pick up an animal, immunize them over the course of, let's say, 2, 3, 4 months. You immunize them with your target, and then eventually they will respond to the target and they will produce antibodies against your target. And then you can just purify them, affinity chromatography, and you've got them. If you want to work on something more specific, if you want to have single clones of antibodies, then you need to do something that is a bit more laborious. And so you can start from an immunization, and then what you do is you collect the blood, you collect the B cells, you extract the RNA from those cells. Then with specific primers, you can amplify specific genes. So for example, the VH and the VL encoding genes. And then you can clone them in a vector, generate libraries of antibodies. It can be Fabs, it can be single chains. I'm sure you heard of this in the lab. And then you enrich your library against your target, and this takes months, years, until you get some clones, you express them, and then you test them, an ELISA, for example. You can also do this without relying on animal immunizations. Nowadays, there's hundreds of different types of libraries. You can buy libraries. You don't have to make your libraries from immunized animals. You can use naive libraries where you don't really immunize the animals. But this is just the basics of all the work that I've done in the past. So in my PhD, so after technical, I moved to the UK. I got a scholarship, for a PhD scholarship. And initially, all I had was this title: Generation of Antibody Against Lipoxidized Proteins for Development of Diagnostic Tools. And I didn't know anything about generating antibodies. We didn't even have the target at the time, so I had to start with the first thing on my list that I showed you previously, that was target assessment. So I started by choosing a target. In my case, I selected a protein. We selected peptides on that protein. And what I had to do was to prepare my target before doing the immunization. So I selected 3 primers. I did the in vitro preparation of the antigen. In this case, I had to perform this lipoxidation, which is pretty much oxidizing the peptides. I had to confirm everything by mass spectrometry, check that my peptides were lipoxidized, and that was the first year almost of my thesis. Then I moved on to the second stage, which is animal immunization, purification of the antibodies, and testing those antibodies. So at this time, we decided to immunize sheep because they were slightly bigger than just rats. We ended up by immunizing four sheep. This is a bit, I didn't really put it in a very nice way, but just to have an idea, I immunized 4 sheep. You have the bleeds before immunization in blue, and then after the first immunization, after the second, after the third, in orange, gray, and yellow. And then you see that eventually they start responding to your target. But in my specific case, because I was looking to a modification on a bigger protein, I was always having immunizations against both the protein and also the modification, because they were polyclonal antibodies. So I was raising antibodies against different epitopes. So what I saw was that not all the animals were producing antibodies against that modification. I had just a mixture that was also recognizing the non-modified protein. And then in the end, I just purified them, I test them. But I saw that I had always cross-reactivity with my naive protein, which was not ideal when you are thinking of developing a diagnostic test. So I moved on to the second phase that I showed you, which is production of monoclonal antibodies. And that's when I started doing a lot of work with construction of antibody libraries, Fab libraries, SCFVs mainly. If you remember, like Fabs are just that little arm of the antibody. SCFV is the smallest part, and then in phage display, it takes weeks, months, and you can do it for over a year and not even getting sometimes good clones. But in this case, what you do is you incubate your library with your target, and then you allow the phages that are displaying your antibody again. You incubate them with your target, then the ones that bind, they stay. The ones that don't bind, you wash them away. And then you keep enriching the good clones that you want for like 2, 3, 4 rounds of selection. And then at a certain point, you reach a point where you plate your bacteria, because this is all done in bacteria. And then what you do is you pick single colonies, and each colony is a clone. And if you are working in a lab, you do this manually, and you pick, manually with a toothpick, 100, 200 clones. If you work in bigger companies, there's robots that do this, and you can pick thousands and 2,000s of clones. So I, at this stage of my PhD, I managed to get some good candidates. I picked up some clones. Then I worked for a few months in multimerization strategies. So I had SCFVs, and we were trying to make, at the time, tandem SCFVs. So we were connecting those SCFVs with linkers, and we managed to improve. In this case, the idea was to use them as capture antibodies. So we were trying to increase the affinity, and we managed to go down to low nanomolar, which was quite good. And then I got to the stage where I managed to develop the pre-development of some lateral flow tests. This was pre-pandemic, so at the time we were not really used to use these devices. It was not so common. So with standards, I managed to do some calibration curves. The tests were kind of working, but it was only like a 3-year project, so I didn't got the chance to test it with clinical samples, but it was kind of working. So this was pretty much what I did in my PhD, and also I did this very similar work in my postdoc. Now at Bayer, I'm doing something slightly more different. So it is a discovery focus group. We also do phage display and antibody discovery, but I'm more onto the after panning side, and I'm working with next generation sequencing. So the idea is that after panning the libraries and after doing rounds of selections in those antibody libraries, what we do is we collect the DNA from each round, and then we sequence those rounds. So for example, if you are doing strategy A, B, and C, you have round 2 and round 3 for each strategy, and then we sequence, and then we can see the diversity of clones that we have in each round. Each color is a clone, and then we see that after 3, 4 rounds of panning, you start seeing the clones that have been enriched for your target. Like for example, here you have the clone that is orange that has been enriched from round 2 to round 3. And what we do is based on this enrichment analysis, we select which rounds we want to proceed for screening. So then we select, for example, round 3 of strategy C that looks a bit more enriched than the others. And then we moved on to manual picking clones like I showed you before. So now what I'm working on is on not using just NGS for this. So after doing sequencing, this is just an example. If we plot in this graph, if you look at it, each dot is a clone. And when we pick clones, usually we pick clones that are on this side, clones that are very abundant, clones that have been very enriched. And if we look at the table, we see that clones that are very abundant and have been enriched over the rounds, like the blue ones, for example, we know that we see that they are very little abundant in round 0 and round 1 and 2, but then they get very enriched, which means that, and then by ELISA, we see that they bounce, so they are good targets. But then if we look at the clones here surrounded by the yellow line, we see that they are very abundant, but they haven't really been that enriched. And then when we do when we express them and when we test them, we see that they are not really good binders. But because they are very abundant since from the beginning, since from even starting panning, we ended up by picking a lot of these clones that we don't really want them because they are just abundant but they don't bind. So there's no interest in picking these abundant clones. So what we are trying to do with NGS is to see if we are missing these clones over here, because there's a lot of, for example, this one is an example of the green clone. There are clones that they are not very abundant, but they are there and they are enriched. Like in here, we see that goes up to 64, which is like a very low value. So the chances of manually picking one of these clones are very, very low. So but it is in fact a good binder. So what we are trying is to use NGS clones we are trying to use NGS tools to see if we can find clones that are here in this top left quadrant, so we don't miss any good clones. So the idea is to do panning, NGS analysis, do some sort of correlation between abundance and enrichment, get those sequences, and then synthesize them. Instead of, because if this is a really good clone and we don't pick it, because we would have to pick, I don't know, 5,000 clones to get the chance to pick one of those, then that would take years or weeks and months. So the idea is to just avoid these rounds of manual picking and then just use NGS to extract the sequence and then synthesize it. So I'm working on checking, so we have different projects undergoing, and I'm checking like all these lists of HCDR3s and doing correlations between abundance and enrichment, and actually check if high abundant and very enriched clones are the ones that are binders or not, and check if there's clones that we are losing or not. And the idea here in this group is to get an antibody drug discovery platform the more digitalized possible. So we are trying to reduce the number of wet lab experiments as much as we can, try to use advanced visualization tools, statistical analysis, predictive models, so we can save time and get novel insights. So we can just, this holy grail is to in the future, we have a platform, we type the target, we say what kind of antibody we want, and then it just gives us the sequence. So that the idea is in the future we can have everything more digitalized, as more digitalized as possible. So that's pretty much what I've got for you, and that's all. That's all. If you have any doubts.
Thank you, Isabel. If you have any questions, you can make it now, or perhaps you can continue.
Maybe at the end.
At the end, okay. So, thank you.
Yeah, thank you.
So I think next speaker, Antonio.
So good morning, ladies and gentlemen. Thank you very much to SBE's organizing committee for the invite. It's been a pleasure also to take part in the previous editions, more as an alumni, and this year as a speaker. It's always great to be here. Today I bring you an industry perspective to the whole MABS universe. I tried to give it a flavor of how platforms, and I will explain what platforms are and everything, how they can play a big role in all of that, and also how bioprocess engineers, such as all of us in this room, more or less, can give a major contribution to the whole journey. Speaking of journey, I don't have a slide about my own journey, but just quickly, so I did my master's here at that time. Even though there was not a choice, it was anyway a very good choice to do biological engineering as Masters of Bioprocess Engineering, as I also often call it. Then I had a short detour to the oil and gas industry that I don't recommend. I came back to biopharma and I did my PhD in Imperial, again in MABS, now in Upstream. And right after, so we are now in 2019, I moved to Switzerland to a company called Lonza. Lonza is a CDMO, Contract Development and Manufacturing Organization. I will also explain what that is, to work on exactly industrializing MABs. So we would receive sequences of MABs, so just at the end of, for example, Isabel's work, and we'd just get that DNA sequence. That's what we got. Or we would get a more mature product, already licensed or pre-licensed, and we would industrialize, so produce at industrial scale. Then in 2020, as you might remember, there was something called pandemic, something called COVID, and Lonza was hired by Moderna to manufacture the mRNA vaccine that some of you might have received. So that was my next project. So then I was involved in the industrialization of the Moderna mRNA vaccine until end of last year, and then I left Lonza. And I'm now a part of the World Health Organization mRNA technology transfer program, where we're trying to establish mRNA manufacturing in low and middle income countries, so developing countries. Very good. So now back to the subject at hand. I divided the presentation into halves. So first of all, I would like to explain to you the whole journey of MABs from the labs to the patients and what industrialization is and what role it plays, and then how platforms, CMC platforms, manufacturing platforms, process platforms, etc., can play in it. And I will have in my slides 3 bioprocess engineers. The green one is more the scientist. The blue one is more the classical bioprocess engineer. That's more or less where I fit. And then we have the red one, which is someone who opted more for a more management kind of role. All are valid and they're all open to you. And my purpose here today is to show you how they all fit in this whole process, in this whole journey. Very well. So how is the journey? The journey is long. As Isabel alluded to earlier, we start with a lot of candidates and we start narrowing them down. As you see, this is all preclinical and discovery and basic research. Then we start clinical trials. We have 3 phases. And then, if you're lucky, we get one of those approved by the FDA, the EMA, or whoever the local regulatory authority is. And that's just development. Then you can start making money. And then you have your monopoly in the market with a patent. Then your IP will expire. And then you will start losing money. So your sales will go down. And that's exactly what happened with Remicade or infliximab. Remicade is just a trade name. So as you see, they had 8 years of development. They had 15 years of market exclusive. Then in 2015, the last patent expired in the EU. And in 2015, the first biosimilar came into market. And that meant a decline in sales for Remicade. And that's what you have at the bottom of the slide. the ticket to this journey is quite high, it's quite pricey. So there is here a distribution of how the costs go, but and that's a 2010 estimate. There's also a more recent estimate by Deloitte in January 2023, so just about 2 months ago, that bringing a map to market costs about $2 to $2.3 billion. And that's, again, not a cheap ticket price. And obviously there must be a return on investment. So, and that's why maps are so costly. Several thousands of dollars per gram is normally a good benchmark price. But the cost of goods, so the manufacturing cost, the cost of production, represent only about 1 to 10%. So it could be 10% if you look at COVID maps, but it's going to be down to 1% if you look at cancer indications where the prices are insanely high. Because the fraction of cost of goods is so low, there is not really an incentive to reduce those costs. At least not in this part of the world, in Europe, in the United States, in Canada, and so on. But this means that the annual treatment with a map can cost $20,000 to $150,000 per person per year. Okay, and this is not a cost, obviously, that everyone can afford. And I wanted to have this slide here. It's a very nice report, by the way. And you can see that the total market of maps is 80% in Europe, States, and Canada, and 20% in the rest of the world. And you can see that we in Europe have access to 120 maps that are approved. Whereas if you look at Nigeria, they have 10. If you look at Zimbabwe, they have 7. So it's a big disparity. And that's obviously something that needs to be worked on. And it's a lot easier said than done, as you can imagine. Speaking of price, I wanted to bring another example of Embrel. So Embrel also had a long development, and it was finally approved in 1998. It was developed by a company called Immunex, based in Seattle in the United States. They finally got their dream. They had a map, not a map in this case, but a fusion protein approved. Except marketing the whole thing is extremely costly as well. It's extremely expensive. So they didn't have money anymore. They ran out of money. And in 2021, Amgen acquired Immunex for just $16 billion. And they still made a lot of money on it. And then they had a very long time of exclusive, and they even had a clinical indication approved after patent expiry before the sales started rather slowly declining. So the Remicade case was steeper in sales decline. So how does industrialization fit in all of this? We have the first phase, we have development, and it ends, I'm calling it the toxicology studies, where we studied the map in animals. And then we have the clinical trials. Before we start clinical trials, the process of the manufacturing process must be transferred into a GMP, good manufacturing practices plant. So it needs to be highly regulated before the product is injected in humans. So at that point, the scientist hands over to the engineer the process, okay? And that's tech transfer and manufacturing support. And that was my job for about 3 years. Or 2.5. And then we start GMP operation, okay? We do a first batch, like I said here, before phase 1 starts. During phase 2 or phase 3, we may need to have a resupply batch. That's quite expected. And latest, at the end of phase 3, the most conservative people do it for phase 3, but latest here, you need to validate the process. You need to demonstrate to the authorities that the process is robust and can deliver consistent quality. Then there's a decision to make. Do I manufacture this in-house, or do I hand it over to a CDMO to manufacture it for me? Okay. I wanted to also highlight that the flexibility to make changes decreases dramatically as also our process knowledge increases, and the GMP requirements or the regulatory requirements also increase. The idea is that development and industrialization are never in the critical path, neither to the clinic, so to start clinical trials, or to commercial, so to supply the market. And the challenge is, how do I do everything that I need to do to have a product that has the quality I need and the quantity I need without ever compromising the timelines of the clinical trials or the commercial launch. And this is where platforms help. Platform is a big word. It's been around in the MABS universe for a very long time. It's nothing more than a standard strategy of doing something. There are CMC platforms. These look at everything, all the activities. That's the example, the main example I bring to you today, because it's the one I think that is further away from you. So I wanted to bring it in so that you have an idea of how it works. Then there is the industrialization platforms where we define the production concept, single use or stainless steel, all of that, documentation, scale-up strategies, analytical methods. And then there's the process platform where we actually think, how are we going to produce this drug? Evidently, I don't want to make it too simplistic. I want to call out your attention that creating and implementing a platform really needs a lot of experience and a lot of expertise. Oops. Okay, so just terminology here. CMC means chemistry, manufacturing, and controls. It's a term from the past, from the classical pharmaceutical industry, but we still use it. And it's basically defining or describing everything together. So from manufacturing, development, drug substance, so the active mAb, as well as drug product, so the final vial that you actually get in the hospital, quality control, submission dossier. So everything is part of the whole CMC universe. Example that I took from websites in the spirit of full disclosure. Selexis KBI, it's an American company. They proposed to you that they would bring your mAb from gene, so you hand them a gene, to IND, so submission to the FDA in 11 months. There's a similar proposal by Wuxi Biologics in China as well. They're growing a lot, and they offer the same kind of package in 12 months. And there is a similar offer as well by Lonza, which I will use as an example, also because it's the one where public information is most detailed. And it's also the one I know better, but that doesn't really matter so much because I can only say so much anyway. And the offer at Lonza is also bringing a mAb to IND in 11 months. So how does that actually work? So every platform comes with an entrance criteria. So a platform that is designed for mAbs, don't try to put a recombinant protein in there because it might not work, okay? For example. Then we start with the cell line development. So we get the DNA, and we need to develop a cell line or a cell line clone or a clonal cell line to manufacture the mAb. That takes about 2.5 months. And we ideally, for a platform, we need to have a host cell line identified that we will then transform with the DNA sequence of interest. And we need to define the whole strategy of selecting the clone, making the transfection, and so on. That needs to be defined ahead of time so that we save time then later for every new product that comes. Then when we finally have that cell line, we will need to do a cell line stability study. So we will demonstrate that the ability to produce mAb and the mAb quality and quantities do not decrease over time as the cells replicate more and more. Then let's next. All right, so when we are in the middle of this at some point, there is a step where we generate pools of clones. They are not yet clones, but they are pools of clones, groups, okay? What we can do is we can take those, you see, only after just about a month, and we can do a pool material run, where we will produce the mAb. Doesn't matter if the cell line is not clonal. They are all producing the same mAb, the same clone. Okay, they're just different clones with different productivities, different qualities. So it's a healthy mixture of everything. But the purpose at this stage is just that our upstream process works well. And when that's done, we go to the downstream. This assumes, obviously, that there are platform processes ready for these two. Now, doing this, what I just said, is risky. It decouples the risk, right? So I can proceed with this while I can also start doing that. But it's a risk, right? Because I need to demonstrate that what I obtain here from the process is similar enough to what I will get later with my final chosen clone, okay? That's already mentioned. So then we have what we call a toxicology run. So that's the material that is produced here is injected in mice, in hamsters, in non-human primates, monkeys, etc. So that needs to be a good quality material. Not yet what we do for humans, but it's a very good quality material. That's toxicology. So then we need the clone. So we cannot do it here. We need to wait until that point. At least now with current understanding, might come. And so then is a dependency between this and that. Also, that material will have to be put in vials, in little flasks, right? So there's always a dependency between the drug product and the drug substance. The drug substance is the active mAb, the drug product is just the flasks. Okay. Then, once we have the clone, we can start working on producing the master cell bank. That's basically a very big room or a very big storage unit with a lot of vials of cells. They're all the same, but we need to have many so that we can make many batches, many lots, okay? To do that, we can start here. Once we have a clone, we start banking the clone. Good. What about if the clone is then unstable? Has happened to me in the past. And then the whole program is delayed by about 3 months. Okay, but it's a risk. But we need to know that there is a risk, and we need to manage that risk well and make sure everyone understands that that risk is there, okay? And also there's an element of trust when you hire a company like that you trust that if they're doing it this way, they have sufficient knowledge and data to tell you that the chances of failure are low, okay? Good, then the GMP, finally, the GMP run needs to have not only the clone for the MCB, but also all the documentation needs to be in place, obviously. And then in the case of the Lonza platform, you also have additional guarantees. So the Lonza guarantees a minimum of 1.5 kilos of antibody to start clinical trials. And other elements that you would ask for are pricing, how experienced these people actually are, how good is the team, what if, it turns out that my project needs some modifications, can you do it, can you not, how much will it cost? So all of that needs to be discussed. And because I'm already over time, I will skip this slide, but message is there's a lot that you can do ahead that is platform, okay? Whenever a new product comes, you only need to do this bit down here. So confirm that the process fits your facility and get your documentation to its excellent degree. In terms of the platform processes, again, I really don't have time, but the idea is to show you that this is all that you need to do, okay, in terms of process development. And what you see now in white is what you need to do for each product. Everything that I have shaded out is what you can do ahead of time. So there's a tremendous amount of work that you can do ahead of time. And that's the secret of a platform, is you have a good platform that you put a lot of money and time and resources in developing, so that later, for every product, you can do it really quickly, as I have just demonstrated. So now the scientists often ask, what if it doesn't work for my molecule? Where the engineer responds, too bad. We can still try to make it better, where the manager says, well, if you pay, we can make it better for you. The scientists also often ask, assuming that it works, what if it's not optimized? And here both will concur that optimization has no role to play in this. Okay, the goal is we need to bring the molecule to the clinical trials and to the people. If it's not optimized, we'll care about that in 10 years or 5, but not now. It really is how it happens, okay? Yeah. So quickly running through, process development and industrialization should never be on the critical path. Platforms allow to reduce the time and the cost that you spend in bringing a drug to market. They are a structured approach. I said that. The confidence that you have in the people doing it is based on your experience and their experience. And I hope I conveyed more or less the message that all of you will become highly qualified professionals. You can support MABs or any other product, in every stage of their development, depending on what you like and on what you want. And that's, I think, my main message here. So I moved, for example, from MABs to mRNA, which no one knew anything about in industry. And, I'm still here, so I survived. And so can any of you. So that's my main message. With that, I will conclude. I left my email on the slides, my personal email. So if you want to reach out and discuss or ask, any other questions related or not related, feel free to do so. Thank you very much.
Thank you, Antonio. And so our last speaker, Cecilia, please.
I'll be very efficient.
Stole some minutes.
So you are okay.
It's fine, it's
No.
fine. It
Oh,
was
fine.
super interesting. I really like to, my question is how you establish the platform, right? But that's to talk later. Because apparently is what gets completely fixed. Okay, so now, where am I? Here. So this is a very heavy presentation, apparently. There we are.
Very nice figure.
Okay, so I'm going to talk a little bit about history as well. Yeah. Now I get in a little bit more pedagogical part. Just a little bit of contextualization, because you know that we always stand, on the shoulders of big people who've made very important discoveries to reach to this stage that we are listening now, that Antonio and Isabel, really nice to know how things are done at a more industrial level. But I think it's also interesting to understand how this all began. So this is the part of academia, right? We need to think about all these things. So firstly, you know that the way the immune system works, and the first idea about how antibodies, were generated, how they operated. There was this, I really like these drawings. So this is from Paul Ehrlich, who was actually the first person to propose, the way that antibodies bind to receptors in cells. So these are the original drawings. They are really cool. And what I find really fascinating about antibodies is that, where we are now, it came from very important discoveries made by many scientists. So I would say that the first Nobel Prize related with antibodies came in 1972, when the chemical structure of antibodies was found, yeah? So do you see this Y-shaped structure that we, like to draw now with these fancy colors, etc., were discovered by these two guys. And I mean, if we think about the tools they had at the time, it was really important how they managed to do it. So for me, then we have a very important Nobel Prize as well for the modern use of antibody technology that came out in 1984, Nobel Prize in Physiology and Medicine. And this, importantly, Kohler and Milstein, as you know, you don't know, but they were two very important scientists who discovered the technology of hybridoma cells. Yeah. So I believe it was Isabel who was mentioning this, that you did this in your work. Well, I'm not sure. Or
I've never done it.
You've never done it, but anyway. So hybridoma cell lines, it's basically a way to immortalize cells, that mammalian cells that can produce antibodies in the lab. And this is how monoclonal antibodies were generated, yeah? And monoclonal antibodies are quite important. Of course, polyclonal are as well, but polyclonal not always work, and Isabel had already an example of this. So usually what people say is that in a polyclonal pool, you might have like only 0.5 or 1% of the antibodies that actually work. So, it's really not very efficient. So monoclonal antibody technology, where you have just one clone for a very well-defined target in your antigen, was really a major breakthrough, yeah? Done by these two scientists, Kohler and Milstein. So basically what you do here is you fuse the spleen cells from an immunized mouse with myeloma cells, and you can create hybrid cells. That's why they are called hybridomas, and they can produce just a single monoclonal or a single antibody type, yeah? Then I also think this was quite important. So a Nobel Prize in 1987. So do you see, it's quite frequently related with antibody technology and immunology. And this was also important because it showed how antibody diversity is generated at the genetic level. And if you don't know that, then you will never be able to actually do any antibody engineering. If you don't understand how this, how you can generate diversity, because in antibodies, the structure, you always have this Y shape, yeah? But the difference is that you have a region where you have the variation, the major variation that is involved in antigen recognition. So do you know the name of these regions? I don't know. Who has ever studied antibodies in classes? Just to know if you all know this or not. Don't be shy. Okay, so you know something about it, yeah? So you have some regions that I've seen a face there saying that they don't know. So these regions here, right at the top, it's where you have regions that are called the CDRs, or the complementary determined regions, and these are the real amino acids that bind to the target, yeah? So this is such a big molecule, it has 150 kilodaltons, but you only have this little point at the tips where the antigen recognition is bound, okay? So this discovery from this scientist was basically understanding how antibody diversity is generated in these variable regions, yeah? So this was quite critical. And finally, and very recently, you have the Nobel Prize, and now it's not in medicine or in physiology, it is in chemistry. The Nobel Prize in chemistry in 2018 was actually for the people who developed the phage display technology that Isabel just explained. And that's the way you most conventionally nowadays develop antibodies, although you still use a lot of polyclonal technology, but most of it is, I think, will go towards the antibody. So how does it work? Okay, the phage display. So Isabel, if I say something wrong, you tell me. Anyway, so these are all drawings that I actually got from, that in the Nobel Prize webpage, you can get all the Nobel Prizes, and they have these really nice drawings to explain to, all the public in general how the science works. So they are usually very simple and easy to understand, even if you are a non-specialist. But basically what you do is you have phages, bacteriophages, and you insert in these phages libraries of DNA, yeah? And these libraries, basically, they will have, let's now talk about antibodies. So you never display the entire antibody, right? You only display parts of the antibody. So let me go back to the basic structure. So typically you will display either a Fab region or these even simpler regions. I think Isabel explained a little bit the single chains that you were told, that you heard. So these are just parts of the antibody where you have the antigen recognition site. So typically what you do is you display at the tip of the bacteriophage. So it's like, if I would be a bacteriophage, my hair is where you would have, the single chain, being displayed. Yeah? And this is how, then I would be basically searching for the best head to fit in my head. Yeah. So the best, the best hair that fits in the best head would be basically the best binder for your target. So this is more or less how phage display works. And of course, you will have lots of phages that do not display a good binder, and you throw them away, and you only keep the good ones. So these people, George Smith and Gregory Winter, they were independently. They've worked on this phage display technology that is now really used. As Isabel said, you have libraries that you can even buy. And you can, as long as you select your target, you can then pen this library and look for binders. It's not that easy, but theoretically it should be, right? Because, you have all the tools in hand. Frances Arnold is also very important, and they collectively got this Nobel Prize in chemistry because she developed this directed evolution of enzymes, which conceptually is very similar to phage display, because in phage display you are like mimicking how the immune system works in generating binders towards an antigen. Okay, so it's like the concept is more or less the same, is that having evolution in a test tube, yeah, and not in animals. So that's why they got collectively the Nobel Prize. Okay, so they have to share it by 3 this time. So nowadays, the market is very big. I didn't manage to get this but you have here the DOI, of course will not get there. But anyway, you have lots of resources about antibodies. This is just to show you that you have so many technologies that came around antibodies. So you have first lots of antibody sources. You can obviously use display technologies. You have phage display that depends on phages that infect cells, and then you need to pick up the colonies. You have also technologies, display technologies that do not need cells, okay, which are called, for example, ribosome display, mRNA display. So you have other ways, and you have cellular display, so you can also use cells to display the binders. You have all the polyclonal based technologies, which is basically you need to immunize animals and make them produce your antibody. And then you have lots of engineering that you can do, and also lots of platforms, as now Antonio was saying. For the platforms for antibody means that you have many designs in terms of antibody engineering. So it's not only the full antibody molecule, you have antibody fusions, you have antibody drug conjugates where you conjugate your antibody to a drug. So now you can really play around. You can think that, the antibody technology is like a Lego tool. You have many pieces and you can really make them in many different ways, and you get different molecules. So the challenge here today was to talk a little bit about the downstream. So the upstream, well, you already have a lot of things about the upstream, right, with Antonio and also with Isabel. I just left this as an extractive process. This is not like the most modern biotech that you like to hear, but the reality is that there are still products in the market that come from plasma fractionation. Okay, so you remove plasma from people and then you separate the polyclonal mixture of antibodies that is in this plasma. Okay, and this is still used today. But most of the antibody market is really in the monoclonal antibody area, okay? Where you have, in this case, a reactive process because you have a biological reaction to produce a very specific antibody, yeah? Using all the platforms that Antonio mentioned. So now just talking about purification. So after you produce your antibody, you need to purify it. You have the established platforms that use mostly, I would say, chromatography, right? Membrane technologies, membrane filtration technologies. But you know that in academia, we are always thinking about other technologies that eventually could be interesting to implement, yeah? So this is just to show you a few things that are being developed. Some of them already have products in the market. And so we have, let's say, the most common one, you have porous particles that you use in chromatography. This is the most standard operation. But for example, nowadays you have quite a lot of monoliths, which are like these cigarette filters. I think it's the most similar thing. It's like a cigarette filter, but you use it to purify. So it's a single cast column, let's say. You can have nanofibers, like they look like tissue really, and they are sort of non-woven fibers that it's because they are very fast. in the processing. So there's also some interesting in these developments. You can do a lot in terms of the ligands that you put in these adsorbents to make them more selective or less selective for your target of interest. You can use stimuli responsive materials, precipitation, extraction, crystallization. And so I would say that I don't know when it comes the time that this is just to generate a little bit of discussion, when any of these platforms can really be part of the platforms that are in place in the industry, right? I think that only makes sense if they really bring a benefit to the platforms that already exist. So if they make them faster, cheaper, I would say that probably time would be the most important thing, no, Antonio? Then cost. We can discuss later. But anyway, so this is just to leave you with this slide. Lots of things that are being done in academia. Most of them, this is a field where people need to work with. It's nice if you can have some feedback from industry, because they usually also bring you some problems. In my particular case, in my research lab, we work mostly at the interface of materials and ligands that you can put in these materials to make them more selective for particular targets. And for me, it's quite interesting, because I've been working with this for a few years, and people always said, oh, no one cares about ligands, because, we have ligands that work. Protein A, in this case, that binds to antibodies work. No one cares about anything else. But now it's like a revival. Everyone is interested in ligands. So things we've done previously, now we are getting, that we published, I don't know how many years ago. Now people want to know, can we develop these, because we don't want any more protein A. I don't know why now, poor protein A. I don't know if it's because of patents or what's going on. But anyway, so it's interesting. But anyway, so I leave you with this. Probably, let's say, there are certain things that are being produced in academia. We try, as I said, to involve always some industrial partners, and most of the times we need to do it even in our projects that we have funded, mainly the ones from the European Union. You need to have industrial partners. So this is also good for us, because we have real problems and we try to address them. And so I think for your future, you will have, if you want to work in this field of biopharma and, upstream, downstream, you will probably get along with the traditional platforms. You might also get to know some of these technologies that, have been developed by academia for a few years, and that could now be interesting also to integrate these platforms, yeah? And that's it. So this is, I could be speaking here for a long time, but I think what is really interesting is now to talk with them. So I will stop here.
Thank you, Cecilia. Okay. Now we have about 15 minutes for questions, so we can change to Portuguese. Mm-hmm. Yes?
Aqui foram fazendo algumas perguntas.
Ok.
As pessoas vão fazer as perguntas por aqui.
Queres que eu leia já?
Sim. Acho que sim.
Vou agora falar em português, embora as perguntas estejam em inglês. Bom, ok.
Sabemos se as perguntas vêm de pessoas que falam português ou não?
I don't know.
A partir de...
Vamos assumir que sim.
Yeah?
Mas as perguntas são pessoas que pensavam que era pessoas que estavam a ver.
Não, são
são lá dentro.
São pessoas que estão aqui?
Sim.
Ok. Então por que é que as pessoas não fazem as perguntas?
Nós fazemos assim.
É assim, ok. Mas a Mafalda Vicente está aqui. És tu, ok. Então a Mafalda...
Perguntas de viva voz. Nós não mordemos.
Não queres fazer a pergunta?
Sim,
Então vá, Mafalda, faz favor.
Portanto, primeiro, para começar, para a doutora Isabel, gostava de saber quais é que são os principais desafios na parte da digitalização, não sei se é assim que se diz em português, a parte final da sua apresentação.
Digitalização.
Sim, eu acho que
para fazer a transição.
Difícil, pelo menos eu só agora nos últimos 2 anos é que tenho andado a trabalhar com a sequenciação. Eu acho que o mais difícil é estabelecer uma plataforma que se aplique, eu que não percebo nada de plataforma, não sei, que se aplique a todos os targets, digo eu. Não sei, porque normalmente nós procuramos, vamos fazer um modelo baseado em, não sei, hidrofobicidade ou baseado em certos aminoácidos, e eu acho que se pode-se aplicar a um target, mas se calhar não se vai aplicar a todos os targets. Mas eu acho que a dificuldade é mesmo essa, é arranjar um modelo ou uma plataforma que se adequa a tudo isso. Eu acho que é difícil. Mas,
eu não sei se posso aqui também dizer qualquer coisa, porque isto era um dos outros slides que eu tinha, mas que estava muito grande. O que é que eu acho que é também o futuro, não é? Mesmo portanto, para o tipo de ligandos com que nós trabalhamos, que ao fim e ao cabo, um ligando não é nada mais do que um binder, está bem? Pronto. Pode ser de origem biológica, de origem química, it doesn't matter, right? É algo que liga a outra coisa. E realmente sim, quer dizer, eu acho que há aqui uma grande tendência da pessoa tentar ao máximo prever, com base num target, conseguir prever qual é que será o melhor binder para aquele target, não é? Isso era espetacular, se a gente conseguisse pôr a sequência do target, target é assim, tiki, não é? E aquilo dar-nos realmente uma previsão de quais é que seriam as melhores sequências de targets. Agora, eu acho que a plataforma vai depender, acho que há aqui várias coisas para ter em conta. Primeiro, acho que há a complexidade do próprio target, ok? Pronto, porque o target nunca é só uma sequência, não é? O target vai ter uma estrutura, vai ter zonas mais expostas e menos expostas, vai ter uma flexibilidade, pode vir a ter, portanto, modificações pós-translacionais.
Às vezes nem é a proteína em si.
Exatamente, portanto, eu acho que começa logo aqui com o problema do target, não é? Para esta questão da digitalização. E depois eu acho que, idealmente, para se ter um bom modelo, o ideal era conseguir-se ter muita informação. Portanto, se fosse possível, por exemplo, nós juntarmos todas as bibliotecas que foram usadas, por exemplo, com single chains, imagina, não é? A pessoa conseguir usar a informação de toda a gente para construir um modelo super robusto, não é? Portanto, um modelo de inteligência artificial que nós conseguíssemos dar todas as sequências para os diferentes targets, ter os descritores dos targets e dos, não é? E dos binders, e desde que o scaffold seja o mesmo, e com isso tentar construir um modelo que conseguisse prever. Eu honestamente acho que é possível, só acho que isto iria obrigar as pessoas a partilharem montes de informação
os dados,
para conseguir ter um modelo muito robusto. Mas eu acho que, quer dizer, com a informação que já, mesmo a nível hoje em dia, não é? Nós temos os AlphaFolds, todos estes métodos que nos permitem prever muito melhor a estrutura dos targets. Eu acho que poderia ser possível.
Eu até acho que sou capaz de existir já vários modelos.
Já, a questão é quão robustos são, porque este tipo de modelos, quanto mais informação nós tivermos, mais robustos eles são.
Essa partilha de dados não é assim tão comum, não é?
Não, eu acho que isso era uma coisa que fazia falta para conseguir chegar a este sonho, não é? Da pessoa colocar o target e ter uma proposta de binder.
Tem que se fazer um projeto para partilha de dados.
Não, eu acho que era super interessante ver isso.
Já agora, só para dar aqui uma chega também, a questão da partilha dos dados é muito importante para qualquer área. E quanto mais nós vamos para data-driven approaches, sejam elas AI ou coisas um bocadinho mais simples, para já perceber o que é que se chama AI, ok? Tudo o que é partilha de dados é essencial, não é? E nós temos uma cultura, assim, os dados são caros. Gerar informação, dados são caros e ocupam espaço, e o espaço é caro.
Sim.
É tudo junto. E ninguém partilha, ou é muito raro ver pessoas a partilhar dados. E o mesmo é válido para qualquer plataforma. Como já vimos, plataforma pode ser muitas coisas, mas o mesmo é válido para, por exemplo, a parte dos processos. É o mesmo problema. Cada empresa tem dezenas de milhares de corridas de dados, montes de coisas que foram metidas armazenadas. Eu diria que é quase impossível conseguir convencer qualquer destas empresas a partilhar os seus dados, muito menos sabendo que vai haver outras empresas a partilhar os dados também. Pronto. Essa é uma parte. Esta coisa dos dados é importante, ligando à pergunta original, que é a questão de saber quais são, quando eu faço um modelo, não é? eu trabalhei em modulação durante uns tempos e há sempre uma questão que é importante, que é quais são as variáveis e quantas é que são. Cada altura, se são poucas, o modelo não é descritivo, portanto não serve para nada. Se são muitas, vai ser mais a confusão do que a informação, digamos assim, por um lado. Eu acredito que isto vai acontecer. Portanto, a questão de podermos fazer isto, uma falta que a tua pergunta implicava, digamos assim, acredito perfeitamente que vai acontecer. E não é preciso ir muito longe. Basta ver que nós temos hoje em dia uma coisa que há uns tempos atrás era um sonho, que é eu poder pôr uma sequência de uma proteína ou de um gene e ter a sequência e ter a estrutura cristalográfica à minha frente. Portanto, vai acontecer. Nós não sabemos hoje como é que isso vai acontecer, ou se alguém já fez, em algum contexto em que está protegido e ninguém sabe cá fora. Mas não tenho dúvidas nenhumas de que vai acontecer sem dúvida.
As ferramentas existem.
Sim, é uma questão de saber usá-las.
Eu agora não consigo ver as perguntas, tens de pôr aqui o dedo. No meu dedo não dá. No meu dedo não dá. Mafalda Arnaud. Quem é Mafalda Arnaud? Então, só se favor, diz. Faz que eu perguntar.
Pronto, a doutora Isabel estava a fazer o doutoramento na parte da descoberta de anticorpos. Para quê? Quais são as doenças que estavam?
Sim, neste caso, aqui, eu depois não expliquei, é muito complexo, mas neste caso são proteínas lipoxidadas, que são proteínas que são modificadas por lípidos, por sua vez já foram oxidados. Portanto, aquilo é relacionado com stress oxidativo. E proteínas lipoxidadas existem numa variedade de doenças, diabetes, como é que se diz? Ia dizer arritmias, mas não é artrite reumatoide, doenças cardiovasculares também. Há uma vasta gama, em todas as doenças há stress oxidativo. Portanto, proteínas oxidadas existem uma data de doenças. No meu caso, eu não tinha uma doença específica, eu escolhi um target que por acaso até era a albumina do soro, modifiquei-a e usei-a como target geral. Mas a ideia de criar um teste diagnóstico onde eventualmente se poderia uma gota de sangue e se poderia ter uma indicação de stress oxidativo. Depois, se isso indicasse, claro que isso não iria indicar diabetes, não iria indicar artrite, não iria indicar uma doença específica, mas o projeto inicialmente era centrado em lipoxidação, em stress oxidativo e a ideia era um teste diagnóstico disso. Parece uma indicação de stress oxidativo.
Eu gosto também de fazer perguntas, posso?
Sim.
Estava aqui com... Portanto, nós queremos produzir e ter anticorpos no mercado a mais baixo custo, não é? Queremos diminuir o custo de forma a podê-los, enfim, serem usados por países menos desenvolvidos. O António falou disso. Mas como é que nós vamos fazer isso? O que é que nós temos que fazer? Temos um procedimento standard para purificação, por exemplo. Proteína A parece que nós tentamos, a nível da academia, tentar arranjar alternativas para a proteína A, mas acho que parece que ela é indestrutível, não é? Porque o processo standard continua e fazem melhoramentos na proteína A, etc, etc. Mas como é que nós podemos baixar os custos do processo? Como é que podemos... É a tua nova função agora, não é? É estar, enfim, tentar diminuir o preço de forma a que seja acessível a toda a gente, a todo o mundo. E também a indústria tem que ganhar dinheiro ao mesmo tempo, não é? Portanto, tem que haver aqui um equilíbrio. Mas como é que nós podemos fazer?
Eu acho que ninguém sabe. Eu vou explicar porque eu acho que ninguém sabe. A sua pergunta vem na melhor altura, na altura mais certa possível. Há 2 semanas atrás, 9 e 10 de março, houve uma reunião muito alargada em Genebra, promovida por uma série de instituições de saúde pública, entre os quais a MPP, onde eu estive, exatamente com essa pergunta. Como é que se vai conseguir tornar os anticorpos monoclonais acessíveis do ponto de vista de existir, porque em português a palavra é a mesma. Portanto, em inglês seria accessible and affordable. Portanto, deles estarem disponíveis, como eu mostrei no slide, há países onde eles nem sequer estão disponíveis, nem sequer estão registados. Portanto, não é uma questão do preço, é uma questão de não existe.
Da logística também, não é?
Da logística, e depois há a questão do preço. Quando falamos em tornar anticorpos acessíveis em países em desenvolvimento, há a questão do processo, e aquilo que eu dizia, os preços do processo, daquilo em que nós nesta sala provavelmente trabalharemos mais, que é a parte do processo de produção, representa uma percentagem relativamente pequena, o que dá alguma esperança, porque significa que o resto é margem. Quando eu digo é margem, vou-lhe chamar, eu não sou de gestão, não percebo nada de gestão, nem muito menos de economia, mas vou-lhe chamar margem bruta. Ou seja, há aqui uma questão que é lucro puro, portanto, eu fiz este produto e estou a ganhar dinheiro com ele, e há uma outra parte que é, eu gastei 2 mil milhões para o fazer. E, portanto, parte disso é abater ao investimento que eu fiz. Mais uma coisa, destes 2 mil milhões não são para um produto. Porque, tipicamente, para um que chega ao mercado, há muitos que ficaram para trás. E, portanto, se considerarmos que são 10 ou 100, há aqui muitos valores no meio que podemos pôr como cenários. Mas há uma parte disso que é definitivamente abater ou tentar pagar aquilo que se envolveu numa altura em que, como a Isabel mostrou, podemos ter dezenas, centenas de candidatos. Pronto, então eu acho que há aqui várias coisas. Quanto à proteína A, é muito engraçado, isso falou-se também lá. Aliás, houve uma apresentação da academia precisamente sobre isso e da importância da proteína A nos custos, etc. E houve duas coisas que foram ditas que eu acho que vale a pena dizer aqui, que é, uma é que a guerra contra a proteína A é uma guerra sinusoidal, não é? Portanto, agora estamos em guerra, daqui a 5 anos estamos em paz, daqui a 10 anos estamos outra vez em guerra. Quando eu estava no técnico, estávamos num pico. Portanto, havia a guerra contra a proteína A, fosse na altura, por exemplo, aqui trabalhava-se fenilbornato e sistemas de 2 fases aquosas como possíveis armas, não é? Como possíveis alternativas. Há uma série de outras. Agora, o que é facto é que, até agora, as armas que foram usadas não foram eficazes. Isto é um ponto. Outro ponto que é importante perceber é, se estivermos a falar de produção em escala, e eu não vou dizer larga escala, porque larga escala não só é muito mais vasto, como o tamanho do mercado, e este é um dos grandes problemas. Nos países em desenvolvimento é uma coisa que ninguém sabe. Ninguém sabe ao certo quanto é que é o mercado, quantas pessoas é que de facto precisariam de cada um destes anticorpos. E sem saber isso, eu não posso, e as pessoas que estiverem aqui no 5º ano percebem claramente isto, que é assim, se eu não sei quanto é que é a minha procura, eu não consigo fazer um projeto. Okay? Pronto, isto é um problema. Agora, para produzir à escala, em princípio eu não vou de repente começar a produzir 20 anticorpos. Ok? Vou começar a produzir um. E se conseguir levar um para países em desenvolvimento nos próximos 5 anos, é uma tremenda vitória de saúde pública. Ok? Pronto. Ao fazer isso, eu vou ter a mesma resina de proteína A que vai poder ser usada muitas vezes. Neste tipo de modelo, que é um modelo de produção mais próximo daquilo que nós chamamos, na Europa e nos Estados Unidos, a produção comercial e não a produção clínica, uma quantidade de proteína A é usada 400 vezes, 400 ciclos, mais ou menos. O que significa que, sobre o número total de lotes que eu vou produzir, apesar de tudo, não representa assim uma fração tão grande do custo. Claro que se puder reduzir, melhor encantado da vida. Agora, há outra área que é muito importante, que é a intensificação. E os monolitos, por exemplo, já estão na indústria. A precipitação já está na indústria. Há coisas que eu posso dizer e outras que não, mas estas posso. E não só estas estão na indústria, como também houve alguém aqui há uns anos atrás, e não foi há muitos, foi há 10 ou 12, teve uma ideia que foi, ok, então e se eu fosse usar uma coisa parecida com o que se usa nas hectares para precipitar tudo o que aparece na água, então e se eu usasse isso para produzir um anticorpo, ou para purificar um anticorpo que sai de uma cultura de reator. E isto está a indústria em GMP, a produzir os anticorpos mais valiosos do mundo. Eu não posso dizer mais nada. Mas isto está a ser feito. Portanto, não estamos... Eu acho que o processo neste momento não é o bottleneck. Há uma questão importante aqui, que é haver vontade. Sobretudo da parte da indústria e também da parte dos Estados. Ou seja, isto aqui é uma espada, não é de 2, é uma espada de 20 gumes. Que é assim, as empresas têm que continuar a ganhar dinheiro, o que significa que não podemos, minha opinião só, não podemos abolir as patentes. Porque abolir as patentes está ok, então está bem, então também não desenvolvo mais nada. Porque não tenho exclusividade.
Não tenho exclusividade.
Mas pode haver aqui uma solução de compromisso, ou várias, uma ligada às patentes, que é dizer, ok, é um contrato, ok, eu dou-te a patente, mas esta patente vem com condições. E uma das condições é, tu vais-me dizer aqui à cabeça qual é o teu modelo para levar este anticorpo para o país em desenvolvimento. Isso é uma hipótese. Outra hipótese que foi feita pela Gilead, para quem não sabe, uma das maiores farmacêuticas do mundo na Índia, mediado pela Medicines Patent Pool, por acaso, foi colaboração. Eles colaboraram, decidiram fazer uma colaboração com uma molécula, uma small molecule na altura, portanto, uma molécula da farmacêutica clássica, e dizer, ok, nós vamos transferir este processo para a Índia, e o que acabou por acontecer foi que a marca da empresa com que essa parceria foi feita representa neste momento uma gama de produtos da Gilead, alguns produzidos por eles, outros não, na Índia e numa série de mercados na Ásia. As estratégias de colaboração podem vir a ser um modelo. Agora, a Gilead fez isto, que eu saiba não há muitas mais a fazer isto. E é preciso ter uma visão. A Gilead fez isto porque tinha um CEO que queria fazer. É sempre assim, não é? Se quem está no topo disser não, é não. Ponto final. Isto é a parte da visão e da vontade.
E da vontade.
Tem muito a dizer aqui.
Claro. Por acaso posso aqui só fazer uma pergunta. Eu acho que realmente nesta questão de levar portanto, este tipo de biofarmacêuticos para estes países em desenvolvimento, não há dúvida que um dos problemas é de facto a identificação das necessidades, não é? E se os próprios sistemas de saúde não têm estruturas e não têm capacidade de fazer exatamente esses rastreios exatamente, não existe, não há uma capacidade organizada de fazer esse mapeamento. Epá, eu também não posso dizer, olha, o que eu precisava mesmo era ter não sei quantas unidades disto, não é? Portanto, é um problema que de facto é mais complexo, não é? Tem a ver com um problema de desorganização.
Da organização, da logística.
Não é mais geral, mas não deixa de ser importante porque, ao fim e ao cabo, ao tentar estar a resolver um problema, podemos também estar a puxar, alavancar a resolução do outro problema.
E é trazer os problemas todos, ok, o problema é o quê? É os custos, ou o problema é não haver um sistema de saúde que é capaz, ou é não haver uma autoridade regulamentar que sequer é capaz de rever um dossiê de aprovação.
Claro.
Porque a droga pode não estar submetida, porque a autoridade regulamentar simplesmente eu sei lá o que é anticorpo. O que é monoclonal? Isto acontece. Ok? Eu não estou a tirar estes exemplos assim. Isto existe. Ou seja, então estes são os problemas. Ok, então o que é que nós temos que fazer? Temos que trabalhar o processo, temos que treinar as entidades regulamentares, temos que capacitar os sistemas de saúde, temos que promover campanhas de rastreio. Agora,
interligar tudo.
E juntar tudo. Isto não se faz num ano, nem em 10.
Claro. E toda a parte das vacinas também.
Tal e igual. Eu estou mais, aliás, nessa parte. Portanto, os anticorpos, eu não estou para já muito envolvido. Mas nas vacinas é um bocado o problema, que é assim, as vacinas da mRNA estavam na academia, confortavelmente, até que de repente apareceu o Covid, que acordou as vacinas da RNA e veio tudo para a indústria, de uma hora para a mão, um grupo de pessoas que não fazia a menor ideia do que era, que sequer era possível fazer vacinas da RNA, de repente passou a produzir à escala mundial. Os maiores projetos estavam a ver.
Mas foi muito rápido.
E foi muito rápido.
Pronto.
E não vamos. Bem,
é possível.
A falar sobre, sim, há uma série de coisas que contribuíram e que foram muito importantes para isso. Agora, a questão é, quando as primeiras vacinas foram aprovadas na altura para uso de emergência pela FDA ou pela EMA, estamos a falar de autoridades regulamentares que têm, alegadamente, as pessoas com melhor treino na área. Agora imaginem o que é agarrar num dossiê para a aprovação de uma vacina da RNA, chegar à Nigéria e dizer assim, está aqui, podes vacinar as tuas pessoas. O que é a RNA? Ok, este exemplo não é real, mas isto existe. Chegar a autoridades regulamentares e começar a falar de coisas e eles ficarem a olhar para nós, o que é que tu estás a falar? Isto existe todos os dias. Ok? Portanto, essa parte é extremamente importante. Extremamente importante. As vacinas igual.
Então eu tenho agora uma pergunta que é...
Estou baixo de fogo. Eu sou proteína.
Não, é mesmo curiosidade.
Não, estou a brincar. Sim.
Se conseguiu, durante a pandemia, se conseguiu o quê? 2 anos? A pandemia durou
menos.
Um ano
foi menos, foi um ano?
Se conseguiu demoraram 4 meses.
4 meses?
Sim, meio ano, se pôs um lote de vacina aprovado.
4 meses, foi menos, 4 meses.
Tenta fazer isso agora para outros candidatos.
Isso é sempre possível, vamos lá ver. Eu não queria desviar nem o tema, mas muito rápido. Há um conjunto de tecnologias hoje em dia, vamos lá ver, o RNA não foi possível chegar aqui e dizer, então agora vamos pensar qual era a melhor maneira de fazer RNA. Não, assim, o que é que existe? É anticorpos. O que é que tens? Tens uma máquina de cromatografia assim, tens uma TFF assado, tens um reator não sei o quê, vai ter que dar. Traz tudo para aqui e vamos ver como é que isto anda.
É partilha de outra vez de informação.
Aí houve muita...
Houve partilha de informação também.
Mas vamos lá ver, houve no modelo de negócio tradicional. Porque quem é que tinha o conhecimento da tecnologia? A Moderna, a BioNTech, a CureVac, outras com um bocadinho menos de escala, ok? Quem é que ia produzir? Era impossível qualquer destas empresas criar uma unidade de produção em 4 meses. Impossível. Portanto, quem é que sabe de produção? São os CMOs. São as Pfizers, a Pfizer não sendo uma CMO, tem uma unidade de CMO, e além disso tem muitas unidades, portanto sabe muito do processo. As Lonzas, a Thermo Fisher, a Catalent, e uma série de outras. A Emergent, que muitos problemas teve nas notícias mais vezes do que gostava. Portanto, estas pessoas sabem de produção? Ok. Então é essas que eu vou explicar como é que isto se faz.
Pois.
E eles que se arranjem como quiserem.
Mas pronto, houve essa interação toda.
Mas no modelo pago.
Pois, modelo pago. Só tenho aqui uma última pergunta, que acho que é para o António, acho que é interessante. Qual é a diferença entre trabalhar numa NGO e numa companhia? Pronto. Acho que é interessante para eles perceberem e para saberem.
É muito, muito diferente. Atenção, eu estou numa NGO há 3 meses, ok? É muito fresco. Mas é assim, numa empresa há uma driving force fortíssima para fazer as coisas. Eu sei que isto parece evidente, mas fazer quando tem que ser. As deadlines são coisas muito rígidas e no orçamento que está desenhado. Se alguma das 3 não jogar, a coisa não vai correr bem. Se não for feito a tempo, ou se custar o triplo, a coisa não vai, ok? Pronto. Claro que é preciso planear, quando eu digo custar o triplo, é preciso que a estimativa de base faça sentido, ok? No mundo das NGOs, a prioridade está no fazer. Se demorar mais tempo, pode ter que demorar mais tempo. A prioridade está no fazer. E isto prende-se um bocadinho com, e posso dizer muito abertamente, aquilo que mais me atrai nesta nova função, que é a missão que está por trás. Que é, se o projeto é arranjar uma forma de fazer rastreios de cancro da mama, por exemplo, na África subsaariana, se eu estabelecer como meta que vou fazer 2 milhões de rastreios em 5 anos, e se não fizer em 10 anos, a missão está lá na mesma e avançou-se muito. Okay? Isto numa indústria era impossível. É impossível eu dizer, dentro do que estou a fazer a documentação, exatamente, que estou a fazer a documentação para estabelecer um processo e dizer assim, ok, isto tem que estar feito neste dia porque o lote começa um mês depois, e eu dizer assim, vai ser preciso mais um mês. Isto não vai acontecer. No mesmo dia eu tenho, das duas uma, ou eu estou fora ou há mais pessoas a trabalhar comigo. As duas não vai acontecer atrasar um lote de produção porque um documento não está pronto. Atrasar-se, eu digo atrasar muito. Okay. Portanto, há esse drive enorme. E depois não há aquela coisa de, então o que é preciso? É preciso uma pessoa que sabe, vamos supor, cromatografia. Uma pessoa que sabe de cromatografia, que tem experiência, que conhece por trás a ciência, etc, e sabe de produção, seja qual for a área. Se estiverem numa indústria, isto não é necessariamente um problema. Ou existe dentro de casa, ou se contrata. Quando vocês estão numa NGO, isto pode não ser possível. E, portanto, muitas vezes faz-se com a prata da casa. Com quem está, todos juntos, estuda-se e vai-se tentar resolver os problemas. Não há a mesma abrangência e a mesma facilidade. E também por isso é que as coisas às vezes andam um bocadinho mais devagar. Aquilo que para mim, agora falando do outro lado, é que eu quando estava na Lonza, e quem está na indústria em geral, na área de produção, não vê os pacientes. Não vê no sentido, nem sequer é tema. Okay? Quem trabalha em produção, trabalha em produção. Eu aprendi o mesmo em 3 anos que muita gente aprende em 10. Aturei aquilo que fiz. Okay? Agora, eu não via os meus pacientes. E eu, quando dava treino às minhas equipas de produção, fazia sempre questão de lhes explicar o que é que eles estão a fazer. É um anticorpo, serve para isto. Daqui a um ano vai estar nestas pessoas. Okay? Porque eu tinha acesso a essa informação. E eles não tinham. E eu acho que isto, eu fazia isto porque eu acho que isto é das melhores motivações que uma pessoa pode ter para fazer o trabalho que está a fazer. No menos eu é ao contrário. A missão está sempre lá e toda a gente a vê todos os dias. Okay? Às vezes nós podemos não ter os skills todos que são precisos. Mas a missão está lá e isso é um drive para mim, e isto nós somos pessoas diferentes, não tem que ser igual para o resto. Para mim é um drive fortíssimo. Levantar todos os dias às 5h15 da manhã para fazer isto. Que é o que eu faço. Acho que tentei mais ou menos responder.
Obrigado. Não sei se querem dizer mais alguma coisa, mas acho que já passou o tempo, já não tenho, não é?
E só dizer que estou completamente disponível. Se o meu e-mail e quem não tem, há na sala mais quem tenha, portanto podem partilhar à vontade.
Sim, mesmo para mim também.
Se quiserem uma vez alguma coisa, digam.
Claro. Então, olha, muito obrigada aos três, foi muito interessante e pronto, e mais uma vez agradecer. Agora tenho que escolher se querem ir mais para a fase do início, do meio ou do fim, lá do chapelinho vermelho. Qual é o verde, o azul ou o vermelho?
Só para finalizar,
Silêncio,
a Inês Cardoso está aqui na sala.